right. Good morning. My name is Carrie Albertin, and I recently completed my PhD working with Cliff Ragsdale at the University of Chicago. And I just want to begin by thanking the organizers for inviting me here. It's very exciting to be here at the dawn of a new technology that promises to open up whole new worlds of biology. Much as the sequencing of the human genome led to a birth of new technologies and tools that have allowed us to sequence now hundreds of animal genomes. And indeed, if we look across the 30 extant animal phyla, we can see that there has been a burst of sequencing in a number of different groups. Particularly, you'll notice that there are more than 300 chordate genomes, mostly located within the vertebrates, such as frogs and fish, mammals and birds, as well as nearly as many arthropod genomes, including the model system Drosophila melanogaster, and some 80 nematode genomes, including that of Caenorhabdites elegans. But if we consider animal diversity as a whole, looking across the animal tree, we can see that the bulk of this work, along with the bulk of model organisms, are restricted to two of the three main animal groups. There are a handful of sequences available in basally diverging animal groups like jellyfish, ctenophores, and sponges, but the bulk of the work has been done in bilaterally symmetrical animals, highlighted here in gray. With much of it found in the deuterostomes, shown here in red, which include the chordates and the echinoderms, as well as the ecdysozoans, highlighted in green, such as arthropods and nematodes. However, you'll notice there's this third group of animals, highlighted in blue, called the Lophotrochozoa. And the Lophotrochozoa contain roughly half of all extant animal phyla, and yet have been relatively underserved by this boom in genome sequencing. So I wanted to take the remainder of my time to tell you one story about one of these genomes found in the second largest animal phylum, that of the mollusks. And so this is the genome of Octopus bimaculoides, or the California two-spot octopus, shown here. And so why study octopuses? Well, I mean, frankly, they're cool. They have a suite of features that are not found in any other animal group, including this crown of sucker-lined flexible arms that surround a beaked mouth, which sits right about here. Their esophagus actually passes up through the center of their brain, which sits in between their eyes, on up to the mantle, which contains their viscera, including 3 hearts, 2 gills, and an ink sac. And of course, cephalopods also have a suite of really remarkable behaviors, including the ability to change the color and texture of their skin to perfectly camouflage themselves to the background. And this suite of remarkable behaviors is governed by their large central nervous system. Octopuses, along with their cephalopod cousins, squid and cuttlefish, have the largest invertebrate nervous systems. And just to give you a sense of the scale of this, here we can see the brain of an adult mouse shown at the same scale as the brain of an adult Octopus bimaculoides, which is a small to medium-sized octopus. We can see here the supraesophageal and subesophageal brain flanked on either side by the large optic lobes that connect to the eyes, which would be on either side of it. This actually is less than half of the octopus central nervous system, with the remainder of it contained in the axial nerve cords that actually run down the center of each of their 8 arms. And so we can see that these nervous systems are put together rather differently, but are a classic example of convergent evolution, which gives us a fantastic opportunity to try and understand how large genomes are put together. And so we know from decades of research in mice that there are a number of gene families that are involved in setting up the vertebrate nervous system, including the protocadherins. So protocadherins are a family of cell adhesion molecules. They have 6 extracellular cadherin domains, a transmembrane domain, and an intracellular domain. And it's thought that the protocadherins, which are expressed in neurons, act at the cell surface essentially as a molecular barcode. They can combine together, mix and match, to create a great diversity of signals at the outside of neurons that help in setting up neuronal circuits in vertebrate brains. Vertebrates have quite a few protocadherins. We can see here the 58 clustered protocadherins found in the mouse genome. So they're found at a single locus in 3 clusters, the alpha, beta, and gamma clusters. And as I mentioned, vertebrates have quite a few of these. So there are 53 clustered protocadherins in the human genome and more than 100 in the zebrafish genome. However, if we look at invertebrate deuterostomes, such as sea urchins and acorn worms, we find a single protocadherin in their genome. Searches in icthysozoan genomes, like those of the fly and the nematode, turn up none. However, one has been recovered in a cnidarian genome, suggesting that the last common ancestor of bilaterally symmetrical animals had a single protocadherin that was lost in the icthysozoan lineage, but greatly expanded in vertebrates. So we were quite surprised to discover more than 160 protocadherins in the octopus genome. In addition, we recovered dozens in other lophotrochozoans, including annelids, snails, and clams. And when we consider the relationship of these different protocadherins to each other, we can see that they cluster based on their lineage. So here in yellow are the octopus protocadherins, and we can see that they largely segregate from the protocadherins found in another cephalopod, that of the Boston market squid Doryteuthis. And we can see that these cephalopod protocadherins are segregated from other protocadherin expansions in mollusks, humans, and annelids, suggesting that the expansions of protocadherins that we see in these lineages were actually independent. In addition, we see that the octopus protocadherins are found in genomic clusters, paralleling what we see, for example, in vertebrates, and presenting both an unexpected role for protocadherins in setting up the largest invertebrate nervous systems, and providing a fascinating molecular parallel for the convergence that we see at the morphological level between cephalopods and vertebrates, and also just providing fundamental insights into nervous system function, as well as a whole system that would greatly benefit from advances in such things as synthetic biology. And so with that, I will happily take your, I'd like to thank my dissertation advisor, Cliff Ragsdale, as well as my collaborators on the octopus genome. And now I'd happily take your questions.
So I guess the obvious question is, what are the prospects for
Well, everything is brand new in this system. So right now, we don't have gene manipulation in this. We don't have, really, we're in the process of developing a bunch of tools for the system, but you could imagine taking, for example, protocadherins, the protocadherin clusters, and testing them in a different system if you could engineer that. Any other questions? Yes.
In the context of synthesizing DNA using these protocadherins genes, it would be interesting to simulate further expansion of those genes and combinatorial power.
Absolutely.
And just test the consequences of an expanded repertoire in this, for example, in this model organism.
Absolutely. Yes.
At the beginning of your talk, you introduced the protocadherins are actually organized in these different groups, such as alpha and gamma in the mouse genome. Mm-hmm. Do you see any type of distinction of how they're arranged when you look at octopus?
No, and so in the interest of time, I didn't go into explaining this, but the protocadherins that are in the alpha and gamma clusters in mice actually show a really unusual property where each of these variable domains encode the extracellular and transmembrane domains of this, of the protocadherin, but they are spliced onto a common intracellular domain. And so this structure is found in vertebrates, but octopus doesn't have this. We find the extracellular domain and transmembrane domain are encoded in a single exon, just like they are in vertebrates, but they each have their own intracellular domain. Thank you.